Hello, everyone, and welcome to this month's Insights and Practice webinar from Microbial Insights, Science in Action, Targeted Methods for Meaningful Insights. I'm so excited to see all of y'all joining us for the session today, which I really think will tackle a major topic in our industry. Presenting for us today is Microbial Insights' own CEO, Dora Taggart, as well as our Vice President of Applied Innovation, Dr. Sam Rosolina. Dora has been with MI since 2001, driving MI to global recognition and influence by prioritizing cross-industry, regulatory, and academic collaboration. Dr. Rosolina heads up our reporting and R&D departments, ensuring that our lab continues to provide the high-quality data and science that our clients have come to expect. Today, we're talking about science in action, connecting analytical output to decisive insight by knowing what tools to use and how to use them. In doing so, we're really pulling back the curtain to dispel persistent myths in the industry, because practicing good science also requires recognizing analytical limitations and constraints, including the limitations of large language models and AI. At the end of this presentation, we will have a 10 to 15-minute Q&A session, so please type your questions in the GoToWebinar control panel, and we'll get to as many of those as we can at the end of the session. Before I hand this over to Dora and Sam, I do want to remind everyone that certificates of attendance will be automatically emailed out tomorrow to all attendees on the stream right now. Finally, this session is being recorded, and the recording will be available on our website as well as on our YouTube channel in the coming days. You can also subscribe to our YouTube channel to keep up with future webinars as well as our MI video series. Now, please join me in welcoming our speakers, Dora Taggart and Sam Rosolina.
Hi, everyone. Thanks for joining. Thanks for that introduction, Hannah. Always excited to be one of the voices on this webinar series because I love all the folks that present on this series over the years. As Hannah said, today we're talking about additional insights that can be pulled from molecular data, and we're going to do a little bit of comparison between certain analyses and also what it means kind of in the current age and the future as AI becomes more and more of a topic. When is it appropriate to use, and when is it a concern? And so one of the things that we're starting to see over the years, and this is probably not a surprise for many of you, but as molecular tools become lower cost over the decades, and as they've become more accessible in lots of ways, we're starting to see a lot more molecular data out in the world. And so here on the right, we're showing an image from the sequences that are being held in these different repositories, public repositories. The blue line is GenBank, which is a database of all annotated nucleotide sequences, and then WGS stands for Whole Genome Shotgun, which is its own kind of repository for genome assemblies. All of these are held under the National Center for Biotech Information, which is housed underneath National Institutes of Health. And essentially what you're seeing here is a true explosion of molecular data, specifically lots of molecular data included in DNA sequences. And so we all know that DNA is extremely important. We're starting to be able to utilize it and leverage it in ways that we haven't in the past, and taking so much information that is coded in this, sequence of life that belongs to lots of different organisms. And so it's exciting to see on one hand. On the other hand, as it becomes more and more common, we're also starting to see it used in different ways, and there are some concerns there that I'll discuss here soon. But essentially, more data doesn't necessarily ensure better insights. When we're talking about sequencing, we need to make sure that we know how and when to use that data as compared to some of the other molecular tools that are available and have been available as well. And so we often say that all of these massive data sets, although they can be beneficial under certain scenarios, they can create a lot of noise and in some ways make it harder to understand what's happening either at a site or within a process or a project. because there is so much data and it might be masking some of the information that you're actually looking for. And so it's really important in terms of whatever project or process you're looking at that you select the appropriate tool. And we have kind of always said at Microbial Insights that this should almost always start with a question. So ignore the tool altogether. Let's assume that you don't know the basis of molecular diagnostics or different tools that are available. It's really helpful for you to step back and say, what questions am I trying to answer at the site? So a lot of our examples in this presentation today are examples within the environmental remediation sector, but a lot of this applies across lots of different industries, and we'll talk about that throughout the presentation as well. So some of the first questions can start with, why are microbial samples being collected? And what question will the data answer? And we always want the goal to be actionable data. So we want to be able to provide you with data that helps you make decisions on the next direction to go at a site or within a project. So do I add something, an amendment? If so, do I use amendment X or Y? Or in the world of corrosion, do I add a biocide? If so, which one do I choose? Do I even need a biocide to begin with? These are really important questions to get started with. And so before I dig into some case studies and use scenarios, I want to kind of take a step back and walk through the tools that many of you know, but is beneficial to go back through anyway. And so let's start with qPCR, quantitative polymerase chain reaction. Many of you know this as kind of the bread and butter of quantitative DNA analysis. For those who are unfamiliar with its use in the sectors that we provide, you've probably heard of its use in the health realm, especially during the COVID outbreak, when qPCR was really the gold standard of COVID detection and enumeration. So one of the things that qPCR allows us to do is actually quantify certain gene targets. And so often we break this down into two realms of gene targets. One is taxonomic genes, so who is there, what name tags are there. We can look for those very specific organisms that we want to look for using qPCR. And then the other is, what tools do they have in their toolbox? What functional genes do they have? Do they have the capability to degrade a certain compound? Do they have the ability to cause corrosion of a certain material? These are all really important. And we have those targets designed to help you not only see who's there and what tools they have, but to quantify those. And that's really important. The nice thing about qPCR, and this will be apparent in some of the case studies, is that there is very good specificity with this analysis. And so when we're looking for a specific target, the other organisms, the other DNA that might be present in the sample is ignored. We're going after a very specific section of DNA. And so what that means is that it allows us to get really low detection limits. Really crucial when low detection limits matter for certain organisms. Now, what about sequencing? There's a couple different types of sequencing. One that is familiar to many of you is the 16S rRNA sequencing, wherein the 16S section of DNA is amplified and analyzed. So it uses these kind of universal primers to amplify this section of DNA that tells us the taxonomy that's present. So it doesn't tell us anything about functional capabilities and functional genes. It just gives us information on taxonomy. And it gives us information on taxonomy across the entire sample. So we're looking at a very comprehensive overview of what microbes are present within a sample. What does the entire microbial community look like? So where qPCR is very specific and quantitative, NGS is much more broad. It gives us an idea of the community that's present. And it's also in terms of relative abundance. So it's qualitative and not quantitative. And that's really important. So it gives us a relative percentage of each of these genus level organisms that are within a sample. So we can't quite get down to the specificity of species level within a sample using NGS, but we can get down to the genus level, which is kind of the gold standard of NGS, is down to this genus level, total comprehensive microbial community survey, essentially. But there's another form of sequencing that is also on the rise, and this is what we refer to as whole genome sequencing, or often called shotgun metagenomics. And the way that shotgun metagenomics works is it is also qualitative. So it gives us relative abundances, but it allows us to dig a little deeper. We can get down to the species level of organisms that are present. And we can also look at all the genes that are present in the sample, so we can get down to those functional genes as well. It does not rely on amplification of a specific section of DNA. And so it gives us kind of an agnostic view of everything that is present, including those tools in the toolbox, what functional genes are there. Again, really important to recognize that we're giving broad understanding of what the community looks like, and that this is not the same as quantitative analysis, and especially not in the specificity that qPCR provides us. So 3 very different tools that give us a lot of information between all of them, and really kind of can help support one another when used appropriately. So the reason that Dora and I wanted to provide this presentation is, I'm trying to think of the nicest way to word this. I think the nicest way to word this is that we were frankly horrified to see that a lot of labs, both commercial and academic, were offering sequencing in a form that essentially promised quantitative output. And if it didn't promise, then it strongly suggested quantitative output. This is important for us to make sure that other people understand, because we want to make sure that we're providing the data that people expect, which is why we're always trying to be as clear and direct about limitations and benefits of different analyses that we offer. So when we were recognizing that more and more labs were providing this sequencing data and either saying that it can be converted to quantitative data, or not providing the context to make it clear that it should not be converted to quantitative data, we got pretty concerned, not just for our own work, but for the industries that we serve as a whole. We want to make sure that people are making correct decisions based on data that they get, no matter where they get that data from. And so we decided to put this information together because we've seen over the years quite a few case studies related to this that should provide hopefully some warning signs against it. So the way that some labs are attempting to use sequencing data is to take this relative abundance and then use qPCR of total bacteria to try and convert and say, okay, if we know the specific quantification of total bacteria in a sample, and we use this relative percentage, then that'll give us essentially the same information as qPCR across the board, which is absolutely not the case. And so as an example of this, here's a case study that we have of a release that was discovered in 1999 in Durban, South Africa. This is a site that USK Consulting has worked on. It's an old industrial site, so it's very complex. There's a lot of contaminants present, a lot of chlorinated solvents, and heavy metals. And so the goal here at this site was to determine if enhanced in situ bioremediation could be a good strategy, which is ideal if the risk is low for such a large site and it can be monitored over time. And so with such a large site and groundwater flow moving towards the coast, the question was, are the right degraders present? And there was also a pilot study injection of some amendments that were being tested. We won't get into those within this case study, but the idea was to see, can we both afford this in situ bioremediation infrastructure, which was assumed to be about 25 million in expenditures, and then an additional 1 million annually, kind of indefinitely until the risk was completely removed. And so they wanted to turn to molecular tools to answer this question, which was the right decision. They wanted to reduce the risk of some of this off-site water receptors like the coastline, and then try and move this site towards closure. And so they turned to molecular biological tools to do so. We know a few things about the site. We know that there's a mix of chlorinated compounds present, a lot of chlorinated ethenes, certainly. As I mentioned, heavy metals, which we know can cause inhibition of some of these reductive dechlorination processes. And so it's important to quantify the organisms and genes that are related to these processes because there might be inhibitors present. We know that there's also chloroform, which is also shown to be an inhibitor of reductive dechlorination at certain concentrations. We know within the geochemistry that the reducing conditions are moderate, and pH is good for reductive dechlorination, but low in certain areas. And so what they did was they started by turning to next generation sequencing, and this was performed by a different laboratory than ours. And through that next generation sequencing, they were essentially attempting to quantify what organisms were present. Specifically, they wanted to look at Dihelicocoides. So they also looked at an additional multiple line of evidence here, which is the compound-specific isotope analysis. We have a few webinars saved in our my webinar series about CSIA if you want to learn the basics of CSIA. But essentially, what we're looking for is this number, this delta value to increase over time or down gradient. And so they did see that for certain compounds, there was isotopic enrichment, which indicated degradation of certain compounds, specifically cis-DCE in one of the zones, and they also saw vinyl chloride enrichment in one of the other zones. So they saw some indication of degradation, but it was relatively limited. And so by turning to the DNA analysis, the microbial analysis, they were hoping to answer, why is this limited? And again, as I mentioned, they used 16S or next generation sequencing from a certain lab, and that lab suggested that they sample 17 wells. As it turns out, 8 wells didn't produce usable data, so it didn't provide them with beneficial data that could be used in any sort of actionable way. The results that came out of the next generation sequencing from this lab conflicted with the compound-specific isotope analysis, and it didn't explain what they were seeing at the site. So when we came into the picture, what we suggested was resampling all of these wells and analyzing them for qPCR. As I mentioned, qPCR being very specific, being very targeted, and of course being actually quantitative for these locations. We already know what compounds we're trying to degrade. There's not really a reason to look broadly at the entire microbial community when we know the key players related to chlorinated solvent degradation. And so what these samples were sampled for Dihelicocoides, among other targets related to chlorinated solvent degradation. So let's look at the data. This is starting with the NGS that was run by the other lab, next generation sequencing, giving us a broad identification of what organisms are present. And these are the numbers that the lab came up with after converting using that total bacteria qPCR target. And so after using that target, trying to convert to quantitative numbers, and you can see cells per mil over here on the left-hand side. This is really, really crucial to understand that there were a lot of detections, but again, as you'll remember, 8 of the 17 wells had no detections, didn't have usable data that could help them. So here we see detection of dihelicocoides through these analyses. But then if we take a look at what the qPCR provided in the resampling, we see a very different story. So in some of the locations, we see relatively similar abundances of dihelicocoides in these locations. Wells 79, 18, well 42, well 40, these all look relatively similar. We often say that when it comes to qPCR, we're talking about an order of magnitude difference being a sizable change. So if you're in the same order of magnitude, we can say that these are relatively consistent with one another. But then the question comes to some of these other locations. In some of the locations, dihelicocoides wasn't detected at all, whereas in the qPCR target, we do see detections. This is really crucial when we're thinking about ways to biostimulate for, especially for an enhanced in situ bioremediation project. You want to know what organisms are actually present already that can be stimulated. In some of the locations, they're extremely overrepresented by the next generation sequencing. Well 44 and well 78 show extreme overrepresentation. And in some of these, we see moderate overrepresentation, but it's an issue, right? As many of you know, dihelicocoides has a threshold that we know of that tells us that if it exists at 10 to the 4 cells per mil or higher, then we have a much higher chance, over 80% chance of complete reductive dechlorination. This is based on quite a few publications and has been backed up by the Microbial Insights database, wherein we see ethene production much higher percentages for the locations that show dihelicocoides in this abundance. And so if we know that we're looking for DHC at 10 to the 4 cells per mil or higher, but we're getting mixed signals here, this is really crucial information. Are we at the location where we see complete reductive dechlorination, which is suggested by the NGS data in wells 7 and 6? But in reality, what we're seeing in the qPCR is that these locations are under that threshold that we're looking for. Really important information. And then on top of this, we see pretty good detections in the 8 wells that were previously unusable. In one of those wells, we see no detections of dihelicocoides, but in the others, we see that it's present, which is a great sign. And in some situations, we see above that 10 to the 4 cells per milliliter threshold. Now, we'll talk about this a little more in depth, but the reason that this is that when we're talking about sequencing, as you can imagine, with any type of analysis, you can think of the same thing when it comes to chemical analysis. If you're looking at a very, very broad approach, if you're looking at lots and lots of different, we'll stick with the chemistry analogy, if you're looking at lots of different chemical compounds simultaneously, the detector has limitations in that analogy. So you're limited to what the limit of detection is in terms of each of those individual compounds. The same is true for sequencing. There's kind of a certain capacity that can be reached across all of the different organisms that are present. And so the more organisms that are there, the limit of detection for each of those will essentially increase. And so it can mask the presence of what is actually present in those samples. And when you're looking for targeted, enhanced in situ bioremediation strategies, you want to know in each of these locations exactly what organisms are there, what their functional capabilities are, and how many are there as well. And you're missing that picture in a situation like this. And so what was learned from this very complex site is the historical data is really crucial to provide more understanding of what might be occurring. Again, knowing that there is low pH in some of these wells gives us a little more insight into why we might see lower numbers. Knowing that there's high chloroform in some of these locations is really crucial as well, which is where the multiple lines of evidence approach comes into play. And then also collaborating with experts. Now, some of this is preaching to the choir because many of you who are not labs or consultants with high understanding of molecular biological tools, you should be able to rely on these labs that are providing this information to you, and you should be able to trust that what they're telling you is accurate. So it can be a little tricky. What I recommend here is that you ask hard questions, ensure that the data that you're getting, no matter what laboratory it's coming from, is actionable, and ensure that it's answering the questions that you provide. And so what we always try to do here, of course, is provide that information up front. And of course, this webinar series also provides that information as well. But it's important to collaborate with experts like those in laboratories to truly understand what that data looks like and how it can be best interpreted. Another case study that we have is, again, another complex site. This was another chlorinated solvent site, primarily chlorinated ethanes that were present at the site. And this is another one where they wanted to look into an amendment injection and whether that amendment injection would provide the degradation that they would hope to see, and whether it would actively stimulate the organisms that are present. Now, this is a slightly aerobic site. Determine if by adding electron donors or anaerobic along with the injection of, or not the injection, but the formation of iron sulfide, so the biogenic formation of iron sulfide. So at this location, both NGS and quaternary chlor were provided by us at Microbial Insights to answer some of the questions that the client had in terms of these injections. First of all, will the injection work? And this was another pilot study injection to see if the injection works, and if so, how long does a single donor injection provide biostimulation? And so what they did, which is the proper way to do this, is they did a pre-injection baseline sampling. So who is present under natural conditions without any kind of amendment addition? What organisms? We can look at the entire microbial community using next generation sequencing, and then we can look at those very specific targets using quaternary chlor. And as a reminder, quaternary chlor is essentially qPCR, but it's an entire suite of qPCR targets, in this case related to chlorinated solvent degradation. So instead of just looking for dihelicocoides, for example, we will be looking across the board at lots of different chlorinated solvent degraders and then quantifying them using that qPCR technology. So they did a baseline study to see what organisms were present and what functional genes were present, and then additional sampling every 6 months from time point 1 to time point 5 to see how the donor injection holds up over time. So we mentioned at the beginning that NGS or any kind of sequencing provides a lot of data. This has pros and cons to it. One, of course, is storage. Another is that it can be a little overwhelming when you have lots of data. But the pros are that there can be a lot of statistical analyses that can occur with this data. A lot of additional information can be garnered from it when it's handled appropriately. And so one of those tools, one of those statistical analyses that can be performed is something called principal coordinate analysis. Many of you know about this analysis or have learned about it through our webinars in the past. Essentially what this does is it separates these data points based on dissimilarity. And so it's taking lots of variables and it's collapsing it into 2 dimensions along 2 different coordinates. And then it's forcing those points onto the plot based on dissimilarity rather than similarity. What that does is it gives you some really good indications of which of these are actually similar, because even though they're finding the most dissimilar separation, you still get clustering of some of these data points. So as an example of this, we see at time point 0, a very different location in terms of where its quadrant is in this PCOA coordinate compared to 6 months after injection. The most different that it shows up to be in this entire PCOA over those time points. So immediately what that tells us is that the community as a whole shifts drastically after the injection. And that's what we would hope to see. We would want to see that the injection change the microbial community because that's the goal. But of course, then the next question is, how did it change that microbial community? Was it in a way that enhances the biodegradation of the chlorinated solvents and also enhances the potential for biogenic iron sulfide formation so that there can be additional abiotic degradation of these chlorinated solvents as well? And so what we see over these different time points, and you can again see this visually, you can see that there's a big shift in these microbial communities. So each of these colored bands is a different genus of microbe that shows up in these samples. And what you see in this first band at time point 0 prior to the injection is that a lot of these organisms are aerobic or can use oxygen. And then as soon as you see that injection, you see a massive shift. There's still a small percentage of aerobic or facultative organisms, but a lot of these are starting to shift towards anaerobic. We're seeing sulfate reducers, iron reducers, and fermenters. And from there, we see relatively smaller shifts, but overall maintaining this anaerobic community of organisms. A lot of these organisms are, one, fermenters, which provide hydrogen for a lot of these reductive dechlorinators, but then also iron and sulfur cycling organisms that can produce these iron sulfide minerals that help degrade these chlorinated solvents over time as well. So we can get even more granular, of course, when we look at the qPCR. So both of those previous plots were from sequencing. They give us a broad overview of the microbial community and how it shifted after these injections. But the qPCR, specifically here, the quantarray chlor, provides quantitative data on these clear gene targets that we have. And so these are all chlorinated solvent degraders, or in some situations, functional genes related to chlorinated solvent degradation. Remember that I mentioned that the chlorinated compounds were primarily chlorinated ethanes, and so you see a lot of chlorinated ethanes in high abundance here. So let's walk through the time points. At time point 0, prior to injection, we see essentially non-detects of all of these organisms and functional genes related to the potential for chlorinated solvent degradation as a whole. Now, that's not a huge surprise because, as we showed, these were all aerobic degraders that were present, or aerobic organisms that were present prior to degradation. And then we're looking at these reductive dechlorinators. So we wouldn't really expect to see these present, at least not in high abundances, when we have the system mostly aerobic. But after that first injection, after the first 6-month time point, we see a drastic increase of a lot of these organisms related to chlorinated ethene and ethane degradation. And again, most of these organisms use the hydrogen that is eventually produced by these fermenting organisms as well. So the more fermenters that are present, the more electron donors that these organisms have to essentially eat, and that way they can respire the chlorinated solvents that are present at the site. So the first question, of course, was, did the injection work? I think we can say without a doubt, based on the NGS and this quantarray chlor data, that the first 2 time points show us that the injections absolutely worked. The overall population shifted anaerobic. And not only that, but we see actual quantitation, high quantitation values of these chlorinated solvent degraders that are actively present at the site. So then the next question is, after 1 injection, how much longevity do I get at this site? And we can see over these time points that 2 years pass and we still see some of these organisms present. Now we start to see them decline, but it gives an idea that the 2-year mark is where we start to see that drop off in some of these organisms. And maybe that's about the time that the injection is re-upped across the site after this pilot study injection. So this was a great example of combining NGS and qPCR in an appropriate way, where they both answer different questions about the site and what may be occurring there, and whether or not this strategy moving forward would actually work and for how long. So this is what we're talking about when we specifically say we want to provide actionable data, which is data that helps you answer those questions and move forward over time. So we talked about NGS, we talked about qPCR. Let's talk a little bit about whole genome sequencing. So as I said, when we're talking about NGS, which is 16S rRNA amplicon sequencing, we're talking about a tool that amplifies the 16S region of the DNA, which tells us specifically taxonomy of these microorganisms down to the genus level, reliably down to the genus level. What whole genome sequencing does is through the sample prep, all of the DNA is chopped up into lots of little bits, and then those DNA are pooled together and they're analyzed and recombined to provide complete or semi-complete genomes of the organisms that are present. And within those genomes, we have both taxonomic information as well as functional information. And so we get the taxonomy, relative abundance, and we also get functional genes that are showing up within these organisms as well. So what it does is it surveys the entire community. The other nice thing about whole genome sequencing is it is taxonomically agnostic, meaning that within one analysis, we can look at bacteria, we can look at fungi, and we can look at archaea all together. In this way, it's more cost effective than NGS, because NGS, we would have to have different profiles run separately for each of these taxonomic groupings. It gives us that information about the tools that are in the toolbox and what the community has the potential to do. And it also is really important for looking at emerging contaminants. For example, we just mentioned in qPCR, we know the primary targets for reductive dechlorination. So it's a great strategy using qPCR for reductive dechlorination because we know those targets and we know what to quantify. In this scenario, if we have emerging contaminants or potential pathways, less information that qPCR can provide, and so we can turn to sequencing instead. And then of course, we have lots of data that we can pair with statistical analysis or other omics, for example, proteomics, to really dig into actual biomarkers that might be present. And so I'm showing you some examples of Microbial Insights, whole genome sequencing, and our sequencing includes a pipeline that we're really excited about that uses information from over a dozen different databases to pull accurate information in terms of taxonomic classification, in terms of functional gene annotation, so that we're giving you accurate information through this process. So 2 examples here from the same sample. One analysis, we can look at the bacterial relative abundances down to species level. Again, whole genome sequencing provides down to species level, and also the fungi that are present down to species level. We can also directly compare those between one another, and this helps with that actionable data information. Is there one sample that was treated with something versus another that was not? Or are we looking at time points to understand how things may have shifted over time? What are the environments for each of these? For example, if we're looking at a PFAS site, we take a background sample and we can compare that directly to high concentration and low concentration locations on the site. This gives us a lot more information to work with when we're trying to identify potential pathways. And then again, that functional gene data is really crucial as well. And within our reports, each of our samples will have a long list of functional genes that were detected for that sample, and then codes that can give you a lot more information in terms of database lookup and references for each of those functional genes. On top of that, if you have more than one sample that's analyzed, we can also do these heat maps that tell us in relative amounts in terms of those detections of those functional genes, which of these samples has higher relative abundance of those functional genes compared to the others? And what does that mean in terms of the multiple lines of evidence occurring at your site? So a couple things that are really important to understand about sequencing and its limitations are things like amplification bias. So we just talked about some cool benefits with whole genome sequencing, but what are some limitations that we want to cover quickly? One of those is amplification bias. It is not affected by qPCR. It's not affected in whole genome sequencing, but it is affected in NGS, which again is 16S sequencing. The reason why is, as I mentioned previously, universal primers are utilized for next generation sequencing. And those universal primers, even though they're made to cover as many organisms agnostically as possible, isn't always perfectly agnostic. And so in order to do that, we sometimes overrepresent or underrepresent certain taxa based on which taxonomic segments these universal primers work best with. And so that can shift probability in one way or another. We don't always know what that shift is. And this is one of the reasons that we can't rely on next generation sequencing to provide us with any form of quantitative data. And then I mentioned this briefly, but in terms of DNA sequencing as a whole, both NGS and whole genome sequencing can suffer from low abundance organisms being missed when there's high biomass in a sample. So or across other samples. So when we run samples together, there's a limited number of total reads that are divided equally across all those samples. And so if you have really low biomass, there's a good chance, as we saw with that first case study, that key targets that we're looking for, like Dihalococoides, might be missed if there are lots of other organisms that are around. And so it greatly reduces the chance of detecting some of these low biomass organisms that are present within those samples as well. So with that, I want to pass the mic over to Dora, who's going to talk about kind of advancements, things to look forward to, and what the future might look like in terms of AI as well.
Yeah, thank you, Sam. Okay, so everyone, as I wrap up today, you know, as Sam was talking, we've spent a lot of time and energy over the past year really thinking about genomic analysis and how do we move forward for the future, because we understand that this functional capability and understanding these metabolic pathways is really the future of where we're going to be. And especially as we start to learn more about AI and machine learning, bioinformatics is becoming more a part of genomics. And so, you know, over the past year, we've invested a lot of time and energy and money in creating this proprietary pipeline that Sam was just discussing for our whole genome analysis. And so part of that is getting good alignment of our reads so that we can have the best, you know, analysis that's possible in those reads, so that we can match them the best way possible, so that we can start to understand, you know, the identification of the organisms in these databases. And as Sam mentioned, the reference databases that we use, then it maps to over a dozen different reference databases to be able to identify these organisms, and it annotates all of these different metabolic and functional potentials. And so that has taken, you know, a lot of work to be able to accumulate all of that information into one centralized location so that we can have the best access to that. And then we can use the statistical analysis that he was talking about, like the PCOA plots, the hierarchical clustering, Titan-based analysis, to be able to do better interpretation, looking at samples comparatively over sites across time, and really give much better information, not just genus and species level comparison, but what are they truly doing at the site in that assessment. So if you can go to the next slide, please. So Sam has shown you a lot of that in this analysis today, but I want to zoom out for just a moment and talk about where AI fits into this picture and where I think it's really going, because there's an enormous amount of potential that we're starting to see. And for those of you that have worked with us for a long time, we all know that typically the tools that we get in environmental come from where the money is generated, and that's typically on the medical side of things. So we've seen that the medical industry has really made a lot of advancements in cancer research and drug discovery. And we're actually even seeing really cool opportunities that have come from the mining industry because of the technological advancements that are happening from rare earth element discovery. So these are, coming from AI, these advancements are happening in the genomics sector because of the medical funding and the technology funding that's happening. And with all the samples that we've had come in over the years, we definitely have this extremely large and complex biological data sets that Sam was talking to you about that have been generated from qPCR. We have over 250,000 samples for qPCR, for next generation sequencing, for whole genome sequencing. So AI is certainly going to play an important role for us to be able to make sense of this complex data set. And AI is going to help us to see patterns and understand relationships in this complexity that can be used for better modeling and better future predictions. And one of the more exciting opportunities for us is that researchers are going to be able to use AI for better biomarker discovery to identify new key organisms, new functional genes that, like they're seeing in the medical industry and the mining industry, are going to drive processes like biodegradation. I'm hoping that, we've been looking for understanding the biomarkers for anaerobic benzene degradation for 15 to 20 years. I'm hoping tools like AI will help us to identify and figure out those organisms so that we can then, once researchers identify those targets, what we can do is bring it back to quantitative tools like qPCR or quantarray so we can generate actionable data. And as you know, that's what we try to do here at Microbial Insights, is take those advanced tools and ground them in data that supports real decisions at real sites. So AI, while it's still primarily a research tool, it's powerful. And right now, it can't be independently used for critical environmental decision making. But if you'll go to the next slide, Sam. In order for us to move it to something that we can do more independently, we really need to think about the key risks, and then we can talk about the benefits. So I want to start by talking about the risk, and that's something we think a lot about here, and I think Sam's presentation today really showed that, because it's a simple principle. If you start with garbage in, you're going to get garbage out. And in environmental microbiology, input quality really starts long before data ever reaches this computer. It starts with how samples are collected in the field, how they're preserved, how they're handled even before they arrive at our lab. Then it continues with DNA extraction, sequencing, and ultimately bioinformatic processing. So as you saw in that first case study, if the lab can't extract well, and so we lose 8 out of 17 samples, you're not going to have good data, so you get garbage in. So we have to make sure we have good standardized methods, robust quality controls at every step of the process, because when we have errors like this, those quickly scale in our databases and they lead to establishing misleading conclusions. So we have to consider technical artifacts. If you have bad assay design, so we're creating erroneous data, we get amplification bias, noise, platform variability. All of this can especially come into play when we're talking about things like sequencing data. And if we don't manage these factors very carefully, we'll end up interpreting signals that really aren't biologically real, and they can definitely have a direct impact on our remediation decisions. If you'll go to the next slide, Sam. Even when we have good, high quality data, then interpretability can still be a big challenge. Like even when we have all this good data, our database has these 250,000 samples that I trust the data because I know we produced it well. AI is still, when it looks at the patterns, it can be very effective at identifying a pattern, but it doesn't always explain the patterns in a way that are biologically or environmentally meaningful. We still have the black box problem, which is something we've always had to carefully navigate, and all of you that I've worked with for many years in the field know this. We still have to rely heavily on each other for our scientific expertise alongside data analytics. I've been doing this for almost 30 years, and we have to work together as a team to interpret this genomic data in the context of geochemistry. I need your understanding of hydrogeology and contaminant profiling, understanding the site history and the context of this data. Because without that integration, we're going to simply draw conclusions that don't accurately affect what's happening in the subsurface, and we're going to train these models to give us inaccurate conclusions. So ultimately, the stakes in how we do these interpretations really are going to influence our remediation strategy, our regulatory decisions, and our long-term site outcomes. So what I'm saying here, we really have to take into account training these models with our human expertise, so that it's not an optional part of what we do. It's critical to turning this data into defensible, actionable science. And in fact, it's so important that ITRC, for those of you who haven't heard about this, has recognized this as a critical need, and a team has been formed to help write guidance documents. I've joined this team, and I would encourage any of you that have a passion for what we're going to do with AI and genomics to join this team with me. And let's write guidance documents that's meaningful so we can do a good job of making sure we mold this data into something that can be useful for all of us in the future of this industry. If you can go to the next slide, Sam. Because if we do this well and we work together as a team and we keep these cautions in mind, the opportunity here is really incredibly exciting. We've already started to see this in practice. I mentioned this a little bit in the beginning. We started to see this in medicine and these huge advancements that are being made, but we're now growing in areas that are impactful to us. AI and advanced analytics are allowing us to identify organisms and metabolic pathways that previously were invisible using our traditional methods. We just couldn't get to them. So I'll give you a quick example, and this is one that I personally know well. This is a company called Indolith that we work with. They partnered with BHP, and they developed an AI-guided microbial system for low-grade copper ore, where they bioengineered a microbial consortia that could yield higher copper recovery than they can get from conventional heap leaching. So what this means is they get more copper ore recovery, so a higher profitability from their copper, but also they reduce their reliance on chemical leaching, so they're making a better overall chemical environmental footprint. So it's very exciting from both fronts. And so for me as a scientist, seeing this in the mining industry, it's really awesome. And then the bioengineering that can come from this. So for researchers that are doing this type of work, AI can help them funnel in and hone in on better pathways and not spend so much time and energy and research dollars. It can be a really valuable tool for them. And I think for us in bioremediation, we can begin to collect microbial community data with functional potential and site geochemistry and use it in much more meaningful ways. This directly aligns with the mission of delivering actionable science that we have that helps our clients reduce uncertainty and make much more confident decisions. So for me, I think looking ahead, these tools are going to help us support predictive modeling that can help us anticipate how a site will respond to remediation strategies over time. My goal is to think about how we could use AI and this massive database that we have so we can enable more proactive environmental monitoring, identify potential risks earlier. So while we want to continue to approach AI thoughtfully and critically, I still see it as a key part of the future of environmental microbiology and bioremediation. And importantly, if we can combine this high quality data that we've been producing for all these years with our expert human interpretation, the knowledge that we've gained, I think it's going to give us even better, greater value for the future of what we can do in this industry. So if you can go to the next slide, Sam. So that's to me what actionable data means. When Dr. White formed Microbial Insights, he wanted to talk to the bugs in the subsurface to figure out what Mother Nature had given us so that we could really use precision bioremediation. So I think AI gives us the ability to re-anchor to our core mission, to take all of the information that we have, reduce the uncertainty, take all these multiple lines of evidence together and use it in a very intellectual way to make confident decisions like Sam talked about today, choose the right tools so that we, in a very actionable way, use data very cost effectively, and then use our shared databases, our shared intelligence globally with researchers, with commercial industries around the world to really make better decisions. If you can go to the final closeout slide, Sam. As we close today, I just want to bring us back to a simple but powerful idea. You know, data alone is not insight. Insight alone is not going to give us action. And I've seen firsthand over my career the difference between noise and knowledge just comes down to being intentional. Sam showed in the talk that he gave today that when we select the right molecular tool, whether it's qPCR for quantification, NGS for community profiling, whole genome sequencing, or metagenomics for functional potential, we're not just generating data, we're answering a question with purpose. Because in this era of exponential data, more is not better. Better is better. And what better means to us is targeted methods that align with the decision at hand, making sure we're doing high quality sampling in the field, good assay design in our labs, and then good quality analysis from the very start, multiple lines of evidence working together, not in isolation. And when we do that, we produce something that's far more valuable than data sets. We produce defensible, actionable science. As we look forward to the future, AI will certainly accelerate what's possible. It's going to help us see patterns faster, integrate complex data sets, and it's going to help us uncover relationships that we couldn't before. But I want to be clear, I don't think AI replaces good science, it amplifies it. So if we feed it poor quality data, like we saw in that first case study, we're just going to scale uncertainty. But if we feed it strong, well-designed, high-quality data, we're going to scale confidence, clarity, and impact. And that's where we can stand apart as an industry. We can generate data, we can ensure it's fit for purpose, scientifically sound, and really use it to inform real-world decisions. I think we can create some models that will reduce risk, use the microbes that are there to enable site closure and redevelopment. So hopefully the key takeaway that you take from our talk today that Sam and I really want you to go home with is choose the right tool, generate the right data, apply AI responsibly. And we think when we do this, if we work together, it'll deliver on the promise that actionable data really is what truly changes outcomes for the future. So that's what we have for you guys, and we would be happy to take questions if you guys want to enter questions for us into the GoToWebinar box. And Sam and I are very grateful for you all attending and sticking through with us. I know we're at the hour.
While we're waiting for those questions to come in, I also want to remind that there's 2 handouts available in the communications box. One is a 1-pager on next generation sequencing versus whole genome sequencing, and then some technical details on whole genome sequencing as well.
Sam, one of the questions, can you talk a little bit about the difference in cost of the analysis for next generation sequencing versus whole genome sequencing? Is there a big cost difference between the two?
Well, again, it depends on whether or not you're looking at multiple profiles, which we've found to be the case that many folks do want to look at multiple profiles. They want to look at potential degraders in the bacterial community as well as the fungal community and sometimes the archaeal community. And so when you do more than one NGS profile, it costs quite a bit more than the whole genome sequencing on its own. So in any of those situations, we would certainly recommend leaning towards whole genome sequencing because you get species level detection, you get functional gene detection as well, and then you get those separate list outs of what community members are there within the fungal community or the archaeal community or the bacterial.
Awesome. The other thing too, I guess, do you see AI being used to incorporate molecular data is one of the questions. Like how soon do you see these models incorporating molecular data, like conceptual site models?
I imagine that will happen relatively soon. The biggest concern, of course, is, and I heard someone once say, we hold, so many of you on this call are held to pretty high standards because the work that you're doing is really important. So as a consultant or an engineer, site manager, field worker, you're all held to really high standards because all of the work that is being done is really crucial. And so why would we settle for anything less when it comes to AI models? So I think once we, as an industry, no matter which of the industries you're in, as your industry sees these AI models come through, we all need to be extremely critical, and we need to be really careful about what information is coming out of those AI models and how that matches with experts, because we want it to be held to an even higher degree if we're going to be utilizing them. That being said, I think the technology is moving fast. The concern, of course, is that we're not just dumping this data into an open software, an open AI software, and asking it for output. We really need, as you mentioned, Dora, these models, large language models that are actually being trained on the appropriate data and providing the appropriate output. So once we see that, hopefully that will be relatively soon, but I think we all need to feel comfortable with what's coming out of that black box.
Yeah, I agree. It's an exciting time. Okay, I think Hannah has a question. Hannah, can you read the question since I can't see that one that came up?
Yes, so I have 3 questions on my side, so we'll see how many of these we can get through. One of them asks, how do you deal with the issue of when AI sometimes hallucinates?
We have seen that a lot, and I think that's the important part, is that we're going to have to train the systems and always have the human oversight, because if we don't, we can be sent down the wrong paths. And Sam may have some more to add to this, but we've seen that, and when clients have called into us, and AI has done a fairly good job to get them to us, but it's misguided them or hallucinated or misinterpreted an article that it read. And so it has misguided them in what analysis or what interpretation of what analysis or what it was meant to do, what it said to them.
Yeah, exactly. And I think this is a concern for AI as a whole, is as people take the information that is spewed from AI and they put it elsewhere, like let's say they post that information on the internet without fact checking it, that is then taken back into account the next time a different AI model is asked a question about it. It looks for that information. So the more that happens as well, the more misinformation that is being spread. And so it's really crucial, just as you said, Dora, that we have very specific models for these if we're going to trust them.
We have another question asking about the difference between short read versus long read whole genome.
Yeah, that's
Dora, go for it.
Yeah, that's a great question. And so, it depends on the type of sequencer that you do and the type of reads that you approach and how you kind of chop up the DNA. So, the way that we do it is trying to get as many reads as possible and then kind of piece them back together so that we can get a broad amount of sequencing rather than kind of deep sequencing. So it depends on kind of what you're looking for. So if you reach out to us with the question of what you're trying to do, we can tell you whether the type of sequencing that we do will help in your approach. So if you're trying to look for like a really, really low abundance organism, there may be other types of sequencing that would be more appropriate, where our whole genome sequencing is meant to look kind of at broad-based approach of what all is present at your site.
And there are limitations with each of those as well. For example, some of the really deep reads, the long reads, those can be kind of unmanageable. And so there's typically a higher error rate with long reads compared to short reads.
Yeah, So if you have a specific project in mind, reach out to us and we'll give you kind of honest input of what we've seen. And then some research institutions are better at some of the depth long read, and we can kind of refer you out to those if it's something that's not a good commercially viable process.
Okay, I have one more question that was more specifically about one of the case studies that you covered, Sam, asking about what product was injected in that second case study, if you're able to share that.
Yeah, we're not able to share that. We haven't cleared that with the vendor, so can't go into too much detail on that.
Totally understand. I do have a series of comments that are mentioning that this was a really great talk, really helpful answers, really great insights and input from you guys. So lots of compliments showing up in the chat.
Wonderful.
On these presentations, if you guys have thoughts later, questions, Sam and I are both always accessible. So email us, reach out to us. We'd love to have conversations, feedback. And I'm especially interested in talking to people about AI and especially moving on to this ITRC committee. I'd love to have your thoughts and feedback so I can bring as much value to that committee as possible. So if you have interest in that, please email me.
All right, those are all the questions I have on my side. So I think we will be good to wrap it up here. Thank you everyone for attending this session live. Like we mentioned, certificates of attendance will be automatically emailed out and they'll be sent out tomorrow. And then the recording will be available on the website under the webinar section on our homepage, as well as on our YouTube channel here in the coming days.
Thank you all so much for joining us.
Yeah, thank you everyone. Have a good day.